Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, Bapak-bapak, Ibu-ibu, dan teman-teman semua di seluruh Indonesia maupun yang di luar negeri. Selamat datang di acara pelatihan secara daring, Histology-Based Rapid Reproductive Analysis for Tropical Fish. Pada hari ini, kita memasuki hari keempat atau sesi keempat, sesi terakhir dari pelatihan ini. Saya Eni Buhari, moderator untuk acara ini. Bapak Ibu, pada pagi hari ini kita akan mengangkat topik tentang analisis statistik data reproduksi dengan menggunakan pendekatan R. Kalau hari Senin kemarin kita menggunakan pendekatan Excel, maka pada hari ini tim instruktur yang diawali nanti oleh Pak Ken dan selanjutnya diselesaikan oleh Pak Erik akan mengangkat topik analisis statistik dengan menggunakan pendekatan R. Sebelum kita mulai, sedikit informasi ya, house rules tentang pelatihan ini bahwa mic Bapak Ibu semua secara otomatis sudah dibisukan. Jadi nanti pada sesi pertanyaan, jika Bapak Ibu ada pertanyaan, silahkan angkat tangan. Di bawah ada tombol raise hand, nanti akan dipilih oleh moderator beberapa orang untuk bisa menjawab secara live. Kemudian, jika Bapak Ibu lebih menyukai bertanya secara tertulis, juga bisa mengajukan pertanyaan Bapak Ibu di kolom Q&A yang berada di bagian bawah frame Zoom Bapak Ibu semua. Nanti selama acara berlangsung, kami dari pihak panitia akan mengambil foto partisipan yang hadir di acara ini sebagai bagian dari laporan. Maka kami memohon kesediaan Bapak Ibu foto konsen untuk bisa diambil gambarnya. Jika Bapak Ibu bersedia untuk diambil gambarnya, silahkan nyalakan kamera Bapak Ibu selama acara berlangsung. Nanti panitia akan mengambil foto screenshot dari peserta pelatihan secara daring ini. Berikutnya, di bagian bawah dari frame Zoom juga ada tombol icon globe, yang mana itu berfungsi sebagai simultan interpretasi dalam bahasa Indonesia dan dalam bahasa Inggris. Bapak Ibu bisa memilih kanal bahasa yang dikehendaki, karena selama acara ini berlangsung, kita melakukan penerjemahan secara simultan. Mungkin itu saja sedikit house rule dari kami. Berikutnya saya berikan mic kepada Bapak Dr. Longenecker untuk memulai acara ini. Terima kasih. Ken, time is yours.
Thank you, Eni. Selamat pagi, everybody. Thank you all for joining us again today. I would like to begin with a brief recap of what we did in our last session, which was focused on statistical analysis of reproductive data using Excel. So I'm going to jump right into it with the brief review. And then we're going to come back in a little while and have a series of poll questions that are testing or intended to help us understand your comprehension of what it was that we were trying to show you last time. So I'll jump right into it with Excel. If you remember, we began with me showing you how to copy and paste data into the data template. We then moved on and showed you that for many parts of the worksheets associated with this spreadsheet, data are automatically calculated. However, there were some tasks that you had to do. For the weight-length relationship that I'm looking at here, you had to use the data analysis tool pack to run a linear regression of our log transformed weight and length data. That would automatically calculate for you the A and B parameters of the weight-length relationship. And then we showed you how to plot those data such that you had results that would probably be suitable for publication in a peer-reviewed journal. We then moved on and looked to see whether there was a significant difference in the mean lengths of males and females. To do that, we used a t-test, but first we had to do an f-test to determine whether our sample variances were significantly different or not. And we were looking at this value to determine whether the variances were or were not significantly different. If this p-value was less than 0.05, we would have significant difference in the variance. If it was higher than 0.05, there was no significant difference. So in this case, because we did have a significant difference, we used a t-test that assumed unequal sample variances. Our other option would have been to use the t-test assuming equal sample variances. And again, with that t-test then, we used the p-value to determine whether the lengths were significantly different or not. We then moved on and estimated size at maturity for females. Our big task here was to tell Excel in this cell what the smallest end of the smallest sample size bin was. And in this case, we entered 16 because that was the smallest specimen that we had in the sample data set. Another thing that we had to do to make this spreadsheet work or worksheet work was to begin with a rough estimate of what L50 was. And we just simply looked over here to our observed proportion of mature females. Okay. None of these were calculated until we entered something right here. And I just chose to enter 2.31 because it was close. This value was close to 0.5. We also entered, I believe it was, I chose 29.7 for L95. And we saw that the sum of square values dropped a little bit when I put something in there. But then we asked Excel, using the solver tool right here, to solve for L50 and L95 in a manner that made the sum of squares the smallest possible value that it could. So this was the L50 and L95 values that we then accepted as an estimate for the model that we modeled maturity for the females with. And we accepted 23.8 as the size at maturity, or L50 for the females. And then we showed you how to plot those data so that we could graphically represent what that maturity model looked like. I didn't show you how to do the size at maturity for males, left that for you to do some exploring on your own. But again, you would have to enter the smallest size class here. Again, that would be 16. Once you did that, all of these values would be filled out for you. I'm sorry, through the F column. Nothing would happen in the G or H column until you enter something here. Okay, so you'd look to see what is a value that's close to 50% here in the F column, and then what is a value that's close to 95% also in the F column. When you got that, these cells would automatically fill themselves out, but then again, you would use solver for the cells K1 and K2, asking solver to minimize the value in K3. We then looked at sex ratios. We used a chi-square test to determine whether the ratio of males to females was significantly different. In this case, it was not. And again, this was the overall sex ratio, or the ratio of all males to all females. Our operational sex ratio was the ratio of all mature males to all the mature females in our sample population. And then we went down and looked at whether sex ratios varied with length. And here I zoomed in on the proportion of females in the population, and it looked like the percentage or the proportion of females dropped with increasing size. I'm going to get myself out of the way here, and we plotted a model that was nearly as descriptive as the sigmoidal model that I showed you in the introductory presentation. But you can see again here that this plot shows you how the proportion of females in the population drops with increasing length. We then looked at fecundity, or in this case, specifically at batch fecundity, that is the number of eggs that we expect a female to release the next time she spawned. So for fecundity, Excel went through the fecundity column on the data sheet, pulled out all numbers for fecundity and ranked them in order, then returned for us the log of the fecundity estimates and the log of the length value associated with each of those fecundity estimates. We then used Excel's data analysis toolpack to run a linear regression of those log transformed fecundity and length estimates, and that returned for us an A and a B parameter, which was a model that basically had the same form as what we saw in the weight-length relationship. We had to take an extra step to plot these data because we didn't copy all of the length data over. Rather, we entered a value or a formula in here, which was equals 10 raised to the power of the log length. And I'm going to zoom in here to show you what that looks like, so you can remember what it is that we were looking at. So this was the formula that we entered in this cell. And that just basically back transformed our log of length so that we had just a length estimate. And then we copied that formula all the way down through the column. We then showed you how to plot those results such that you had a length fecundity curve. And again, we explained that typically these curves are a lot messier than what we see with the weight-length relationship, because there would be typically so much variation amongst the females. The final thing that we did then was looked at spawning periodicity. In this case, the data that we plotted were by month. And I believe it's important to remind everybody that spawning periodicity can be examined in a number of different ways. You could choose seasonality. For instance, in Indonesia, you might want to do wet season and dry season. If you are in the northern or very southern portions of either hemisphere, you might look at the seasons, summer, spring, winter, fall. You could look between years if you had a multi-year study, or a very interesting analysis would probably be to do lunar periodicity, where you could convert your date data into lunar phase and then group the data by lunar phase to see whether there was any variation according to moon phase, whether that be new moon, full moon, or the quarter moons. And then again, we showed you how to plot these data where we looked at the mean and the standard deviation in GSI value, or gynosomatic index value, by month. And for our sample data set, it looked like July was the month that had the highest gynosomatic index, or reproductive or spawning periodicity. The one thing that I took special pains to remind you of is that these GSI values, although you will see values here, are not valid until you've estimated L50. So in this case, if you remember, we did estimate size at maturity for the females. So we accept these values, but I had not estimated size at maturity for the males. So these values are not yet correct. In other words, this is including all males in the population, in the sample population, but we only want to be counting the individuals that are large enough to have reproduced or to actually spawn. Okay. So please remember, if you're doing a spawning periodicity analysis using this spreadsheet, you need to estimate size at maturity first before accepting these results. Okay, that is what I would like to show you in terms of the spreadsheets, but now I'd like to take a little time and remind you that we do have some resources available to you in Laulima. I'm going to get myself out of the way. Here in the resources page, we have the standard operating procedure, which has been mentioned various times throughout these presentations. And this really explains the procedures that we're following when we're doing this reproductive analysis. All of our PowerPoint presentations are available in this folder, but the one that I would like to highlight right now is that we also have short videos explaining all of the procedures that I showed you in Excel last time. I'd also like to point out that we do have plotting instructions as a PDF file. The standard operating procedure did not include instructions on how to make the plots that I was showing last time. So if you want a detailed explanation of that, please see this document. Or you can go to this link, which is a YouTube page that we've established that has a series of short videos about every step in the process that I was just talking about, from inserting data to doing a weight length relationship to plotting that weight length relationship. Okay, so if you don't want to read the document, you could go and follow along in the YouTube videos as well. Eric is going to be working on a series of videos, complementary videos that will explain how to do all these analyses in R. So you can check back with this channel in the future and have a series of short clips explaining how to do the R analysis as well. The last thing that I would like to point out then is that last time when I spoke to you, we did not have available an exam for the statistics part of that. That exam is now ready for you. Just go over here and click on the exam for statistical analysis of reproductive data. Click on begin. Just like the one for the gonad stages, you can attempt this an unlimited number of times. If you score 90% or higher, you will receive a certificate that will automatically be generated featuring your name and explaining what the course was about. So if we're ready to begin, you would just hit begin, go into the exam. And here's something that I would like to show you. Some of these questions feature data sets. You will need to go in, use these data sets and analyze them either in Excel or R. We have the answers set up so that you can take whichever approach you are most comfortable with. But to get to these data sets, you'll need to click on the link and they will download for you. And then you just click on your downloaded data and the new data set that you'll be using for that question will automatically pop up and you can copy and paste that into the Excel worksheet, or they're also done in CSV format so that they can be used by R. So they should work in either case for you. Okay. I'm going to go back and show you one more thing before we get out of here. Okay. That is that at the end of the training today, we're going to have a brief poll, but if you would like to instead complete the poll here in La Lima, there's an option to do that as well. This time you're only allowed one attempt because we don't want multiple people giving answers to polls multiple times. So if you would like to take the poll in La Lima, you're welcome to do so here. But again, I believe we're going to have a poll at the very end. Okay, I'm going to take us back to the Excel spreadsheet now, and I have a series of poll questions that we would like to use to see how well people comprehended the information that we were trying to present last time. So, Dendi, would you please launch the first poll? Okay, and as we go through here, I'm going to have to scan or move between files for you to see the worksheet that the question is actually asking about. So for question number one, pertains to the worksheet that is currently on the page or on the data worksheet. Is it necessary to have data in every column of this worksheet to make the spreadsheet useful? And I'm going to set myself a little timer so that we're going to give about 30 seconds per question. I hope will be adequate. Okay, I'm going to move on now to question number two. If I can find my cursor. Question number 2 pertains to the weight-length relationship. So it asks, which data analysis tool is used to construct the weight-length relationship? Okay, question 3 also pertains to the weight-length relationship. And it asks, what is the A parameter for the weight-length relationship that's been constructed here? And question 4 is also about the weight-length relationship page that we have here. It asks, what is the B parameter for that weight-length relationship? Okay, question number 5 pertains to the bimodal size distribution analysis that we did. And it asks which data analysis tool is first used when you're testing for the sex-based bimodal size distribution. Question 6 also pertains to the bimodal size distribution analysis. It says, given the result of this first test that we did, which is the appropriate test for completing your analysis of whether you have a sex-based bimodal size distribution? So given the results of this test, which test did we use over here? Okay, question 7 also pertains to the sex-based bimodal size distribution. And it asked, given that we've done both of these tests now, was there a significant difference in the lengths, mean lengths of males and females for the data set that we analyzed here? Okay, question 8 pertains to the size at maturity estimates for females. And it asks, what is the first task that you have to do when you're using this worksheet to estimate size at maturity? And the final question in this poll is, what value here represents female size at maturity? Okay, and I believe that, okay, Dendy's asked me for a little bit of time here, but as soon as those answers are ready, Dendy's going to post the results for us, and then we will review and see what everybody was thinking. There is a second set of poll questions that we'll use to address sex ratios, fecundity, and spotting periodicity as well.
Hi, Ken. Somebody has asked about question 7, whether or not there might be an error in it.
Oh, I forget what question 7 looks like now.
I think option A should be yes.
Oh, what was it?
Yeah, it's supposed to be a yes/no question, but it has a bunch of text for A.
Oh, okay. Sorry that, yes, there was an error in that. We'll address that when we come to it then. Okay, folks, please just bear with us a little while longer. We're going to have those poll results compiled and then review. Here we go. So the first question is, do you have to have data in every column? About a little over half of you said yes. The actual correct answer is that no, you don't have to have data inserted into any column. That if you have, for instance, length and weight information only, you could use that to do a length weight analysis. Or if you had length and histosex information only, you could use that to do size at maturity. You could also use the same 2 data columns to do to test whether there was a bimodal size distribution. So you don't have to have data in every column for this spreadsheet to be useful. You can do these tests independent of one another if you only have those sorts of data. So for instance, imagine that you were interested in generating a length-weight relationship for a species that you have, but didn't have time or funding to go do the whole set of histological analysis. You could still come up with a weight-length relationship for that species. Okay, question 2 was, which tool was used to construct your weight-length relationship? And 81% of you said regression, and that is the correct answer. We did linear regression analysis on these log transformed data. Question 3 is, what is the A parameter for the weight-length relationship? And the answer, most of you answered, was C, and that is correct. And here is A is provided for you down here in the worksheet. Question B was, what is the B parameter for that same weight length relationship? And most of you answered that correctly as well. And again, right here in the worksheet, we've got it set up so that it shows you that the B parameter is equal to 3.11. Question 5 was, which data analysis tool is the first one used for the sex-based bimodal size distribution analysis? And most people answered F-test for 2 sample variances, and that is correct. We first need to determine whether there's a significant difference in variances so that we can choose the correct t-test. Question 6 was, given the results of this first statistical test, which data analysis tool should be used to test for the sex-based bimodal size distribution? We saw here, and most people said two sample t-test assuming unequal variances, and that is correct. Because this is less than 0.05, the p-value of the F-test, we know that we have unequal variances, so we choose the t-test assuming unequal variances. Oh, yes, sorry about that. So for question 7, we had a little bit of an error there. A should have been yes, and B should have been no. My apologies there. But it's asking, do these results indicate a significant difference in the mean lengths between males and females? So to evaluate that, we'd go down to the p-value of that t-test. And if it's less than 0.05, we say yes, there's a significant difference. So this is 1.59 times 10 to the negative 33, which is very much smaller than 0.05. So we say there is a significant difference in these 2 mean lengths. Question 8 was, what is your first task when using Excel to estimate size at maturity? And most people said enter the lowest end of the smallest size class in cell A2 right here, and that is correct. So if you remember from our last session, we determined that 16 centimeters was the smallest sized fish in our sample. So we entered 16 here. And then basically all of our size classes were built for us in 2 centimeter bins. And question 9 was, what value represents female size at maturity? And that's given for you right here. And most people got that right as well at 23.8 centimeters. So thank you for that. Dendi, could you launch the second poll for us, please? Okay, question 1 refers to the sex ratio worksheet, and it asks, is the sex ratio for all individuals significantly different from 1 to 1? Question 2 refers to the same worksheet and asks, is the sex ratio for mature individuals significantly different from 1 to 1? Question 3 refers to fecundity analysis and asks, what is the appropriate test to use when constructing the length fecundity relationship? Question 4 refers to the same page, and it asks, what is the A parameter for the length fecundity relationship that's been constructed here? And question 5 refers to the same page and asks, what is the B parameter for the length fecundity relationship? Question 6, and this is the last question in this poll, asks, what reproductive parameter must be estimated before accepting the gonosomatic index values on this worksheet? Okay, I believe that completes the poll. Dendi, if you could compile those results for us, please. Okay, and Dendi just sent a message saying that the results are still being compiled. So again, we'll have to wait for just a little while, but we'll come back and see how you all did. I'll get us queued up here on the sex ratio page because that was the first set of questions. Okay, the results have just been posted. The first question was, is the sex ratio for all individuals significantly different from 1 to 1? And here there was a kind of 2 parts to that question. I was making sure that folks understood that overall sex ratio was referring to all individuals. The sex ratio was 1 to 0.92, and the probability value was 0.347, which is greater than 0.05. So in this case, no, this value was not significantly different from 1 to 1. And congratulations, most of you got that right. For question 2, it was the same question, but this time focusing only on mature or more mature individuals, or the operational sex ratio right here. Most people answered yes, and that is correct. Our p-value is smaller than 0.05. So we say that the sex ratio of 1 to 0.76 is significantly different from 1 to 1. Question 3 had to do with fecundity analysis, and it asks which is the appropriate tool to use. And just like we did for our length-weight relationship, linear regression analysis was the analysis tool pack that we wanted to use. So congratulations, most of you got that one right as well. Which is the A parameter for the length fecundity relationship, given for you right here. Most people got this right, 6.36 times 10 to the negative 8. And for the B parameter, most again got this right at 8.35. Again, shown for you right here on the worksheet. And the final question then was, what reproductive parameter has to be estimated before you accept these results? And most people got the answer correct, and that was L50. These values are not valid until L50 has been estimated back here. So in this case, the value for females was correct because we had estimated L50 previously. However, I had not estimated L50 when going through the examples, so none of the male values are accurate, okay? And they won't be until L50 is generated and present in this cell right here, K1 on the size at maturity for the male sheet. Well, thank you all for participating in that poll. I believe we have time for some questions, if there are any.
Yes, thank you, Ken. So now we have apparently 3 people raising their hand. I would like to call Ibu Regina. Silahkan, Ibu. Okay, I cannot unmute this, Dandy. Ibu Regina. Ibu Regina. Oke, sekarang Ibu Umi Khodria. Silahkan, Ibu. Ibu Umi Khodria, silahkan dibuka mic-nya untuk bertanya. I cannot unmute this, Mas Dendi. Pak Andi Syahriana. Pak Andi Syahriana. Okay, now I can unmute it. Okay, so we have 3 people raise their hand, but okay, Pak Agus Arifin, silahkan Pak.
Oke. Izinkan saya bertanya dengan bahasa Indonesia, Bu.
Silahkan.
Ya. Yang saya ingin tanyakan, saya Agus Arifin dari Jatiluhur. Ingin tanyakan, itu kan terkait statistik kan jumlah sampel kan kalau biasanya kan minimal 30 ya, Bu ya. Tapi kadang-kadang kita menemukan suatu jenis ikan yang cukup langka, jadi mungkin jumlah sampelnya kurang dari 30 atau mungkin hanya kurang 10 gitu. Apakah itu mungkin secara statistik bisa diterima atau tidak gitu? Dengan pertimbangan ikan itu adalah langka atau sulit untuk diperoleh di lapangan gitu. Mungkin terima kasih.
Baik Pak, terima kasih. So Ken, the question is about the size of the sample. He said that usually when you run statistical analysis, the ballpark figure for the minimum number of sample is 30. But sometimes he came across situations where samples that he collected less than 30 simply because probably that the fish is a rare species. So how would he address that?
I would run the data anyway. In biology, a lot of people think 30 is a large sample size. In other fields, people don't think that's quite so large at all. You could see from your results whether or not you had a significant difference there. So you can still run the statistical test. If you find that your results are not significant, you may think that basically this is a problem of low sample size. One of the examples I had going through that was for sex ratios. And here you can see, whoops, let me share my screen. Here you can see that we had large numbers of individuals for basically all of the size classes except for the last one. And there had been a downward trend in the proportion of females. But in this case, we only had 1 individual, and that one was a male. And the statistical test couldn't tell the difference from a one-to-one relationship, even though with those patterns, it was pretty convincing that we did have a decrease in sex ratios. Eric, do you have anything to add to that?
Yeah, I think I support your recommendations, Ken. I think that the challenge is with that low of a sample size for things like size at maturity, you're going to have some trouble because the individual data points actually go into the bin sizes. And so the statistic you're working with is a proportion of mature individuals. And even if you've got 30 fish, that often will maybe truncate down into just a handful of those categories. And so it might be tough to identify where that L50 is. But for some of the other tests, say the weight length relationship, you might be able to get a decent fit or a decent model result with 30 samples. But I think Ken's suggestion, just to, in an exploratory way, look at the data you have. It can perhaps help you identify what sizes you may want to go out and try and catch if you have gaps in the middle or bigger size or bigger small sizes. But I agree with Ken's guidance to just go out and do some exploratory data analysis and see what your results look like.
And Eric, just reminded me of something that we've been doing a lot of this reproductive analysis for a long time, and specifically with size at maturity estimates. The least we've been able to get away with was about 125 specimens. And we're always wishing that we had more. So in the case of size at maturity analysis, he's right that 30 probably will not do it.
Sorry. Go on, Ken.
That's all.
Okay.
I was finished. Sorry.
Okay. There is a follow-up question to the to that previous question, actually typed in the Q&A from Pina, and she said that if the sample is not ample enough, for example 30, normally people use non-parametric stat. Do you have any experience applying such a technique, non-parametric?
Yes, we have used non-parametric statistics. Oftentimes, the reason we've done that is because we looked at our data and they didn't satisfy the assumptions of the parametric statistics. And unfortunately, going into the assumptions of all these tests and then also going into non-parametric statistics was beyond the scope of what we were able to accomplish in these sessions. So yes, we do have experience, and certainly non-parametrics are something to look at. Primarily in our case, it's because the data aren't satisfying the assumptions necessary to run the parametric tests. A lot of times, many people say that non-parametric statistics have less power than your parametric statistics do, although sometimes people argue against that as well. So that's one reason why, if you can run the parametric test, it's probably better to do so.
Thank you. Okay, so now we have Ibu Regina. Silahkan Ibu Regina. Ibu Regina. Oke, berikutnya Pak Andi Syahriana. Pak Andi Syahriana. Yang terakhir Ibu Umi Khodria. Ibu Umi Khodria. 3, 2, 1. Okay, so now we are done with the question live, Ken. And the next session is by Eric, the weight-length relationship in R. Time is yours, Eric.
Great, thank you, Annie. And thank you, Ken. Dendi, could you allow me to share my screen, please? So what Ken has covered is approaches for the statistical analysis of reproductive data in Excel. And what I'm going to cover today is a set of methods using the R software for statistical computing that basically will allow us to replicate exactly the type of work that we've covered in the last day and then just reviewed earlier today. So let me get my screens all together. So for those of you on the call that are familiar with R and have used R, just these first couple steps will be very familiar for you. For other folks that have never heard of R, I'm just going to do some quick background so you know what we're talking about. I suggest during this that you don't attempt to follow along unless you have multiple screens set up. It would be very challenging for you to do that. And so I'm going to also share basically a web browser. Sorry, that's La Lima. So if you were to go into a web browser like Google and just type in R software, you're going to get this result, the R Project for Statistical Computing. If you click that, it's going to bring you to the website, and to download the software, up in this upper left-hand corner, there's this download and a link that says CRAN. And so if we click that, so again, we're just looking at how to download this. You don't need to be actively doing this right now. It's just a demonstration of where you would get this software if you needed it. You can click the cloud. So the word mirrors just represents servers around the world that offer this R software for free to download. And if you click that, then you're going to have options. If you have a Linux computer, or if you have a Mac computer, or if you have a Windows computer, there's software versions of R for each of those operating systems. I'm on a Windows machine, so if I click that, it's going to give me the option of the base version, which is what you want. These are the base functionalities of R. And especially if you're installing R for the first time, you click that link. And then you would just click this final link, download R for Windows. That would then download the software directly onto your computer. And once you then go through the steps, it'll ask you to install and basically look at the software. You're going to, for the first time when you open it, see a screen that looks something like this. So this is the R software that we'll be working within. So we'll hide that for now. The other part of what you need, so that's the R software to do the analysis. We need the data. So Ken's already identified this for the Excel, but I'm just going to go over it again. So if we go to our LALIMA website, this is the overview homepage. Off to the left here, we go to resources, and you have this list of all the resources that are available. And if you scroll down, there are two things that we're going to be using. This first one is the CSV called raw data IDLL, and this is that large data set of the demonstration data that we've provided for you that we've been working with in Excel. And then the next is a file called stats in R, and it's a .r file. That's an R script that will be used in the R software. And so you want to have those available. So again, I don't recommend you follow along, but when you have time and you're going to do this yourself, download this CSV file and download this R file. And what I suggest is when you download it to your, sorry, lots of windows going on. When you download it to your computer, that you create, oh, that's not very visible, is it? Let me zoom in for you. So that you create, no, that's not showing up either. Okay, you create a folder. Here I've just called it training within my highest C directory. And within that, I've downloaded the R data IDLL and the stats in R files. And so that's what we'll be working with, that folder for this session. Okay, so let me clean up this a little bit. And now I'm in R. And the first thing I'm going to do is I'm going to open a script. And that's that R script that I just downloaded. So I'm going to go to this training file that I told you about and stats in R. So when I open that up, now we have two active windows in R. The first one that's highlighted is called the editor. The second one behind it is called the console. I'm going to go to this window, just clean them up, tile them horizontally. Actually, I'm going to switch them around. I prefer that way. Oh, mess with me. And so what's going on with these two? The top editor window is where you can write and store your R scripts. And so these are the instructions that you provide to the R software in order to run some of the analyses. The console is where this is actually executed. And as I'm looking, let's see, let's do one thing here. Let's close this again. And let's increase, I don't know why that font went to 10. Let's increase that a bit so we can see better. There we go. Okay, so I'm going to go through that step again of just importing from the training folder the stats in R. Okay. And so this whole process for running these analyses in R is just going to be walking through this script that's in the editor window. And so the first thing you'll notice is that there's a lot of hash marks or these pound signs. And in R, a pound sign represents a comment. So it's not something that's actually executed by the software. It's just something that signifies that there's a note that you want to include. And in this case, this note at the top provides a title, a contact, and it also a URL for the standard operating procedure. The next three comments are just notes, and they basically are to provide some guidelines on what are called hotkeys, so that when you're using both the editor and the console, that you can work more efficiently. So the first note is if you hold down Control and Tab, that allows you to toggle between the console and the editor. And so I'm holding down my Control and Tab, and you can see the different windows are highlighted. And that just means your cursor moves back and forth. And so as you're typing, it's a fast way to just transfer back and forth. And these are little tips and tricks that will help you if you use R. The next is if you want to select lines of code in the editor, you can hold the Shift down and then either use the up or down arrows. And those will then turn blue on my screen. That means they've been selected. And then if you want to run that code, you would click a Control R. And so we've selected that entire control block, and I've controlled our code block. I've clicked Control R. And if you notice down in the console, all of that code now has been written into basically that entry cursor point. Nothing has been executed though, because these are just all comments. And so all they've done is they've been written into the console. And so as we move further down, the first step we'll take is to set the working directory. And if you remember when I was describing where the data is located, I said it was in a folder called training in my C drive. And for those of you that work in Windows, this hopefully looks familiar. This is basically an address on your computer that identifies where a folder or a directory lives. And in R, this command, set WD, the WD stands for working directory. And so for this script where we're going to be running these analyses, I'm setting the working directory to be in this folder that's on the C drive called training. And that just means that when it's looking for files or writing out files, that's where they're going to be. And so it's just good practice to identify where that is. And again, if folks are experienced in R, you may be using RStudio. That's all right. You can use this code in RStudio. I've just chosen to use the standard R GUI just to keep things simple, but this can work in any number of IDEs or integrated development environments for R. And so what we're going to do then is Control R to run that. And if we look down in the console, down here, we see that this just ran. So that means we've set the working directory. And the next step is we want to actually go out and get the data. And once we've set the working directory in that folder, that training folder, now we're going to look for that file that lives in that training folder. And so all we're going to have is the cursor on that line and Control R again. And it writes that out in the console. So now we've set our working directory and we've basically imported the data into R now. And so once the data's in R, we're interested to see what it looks like, right? We're curious if it worked. And so I'm going to go down to the head DAT and click again my Control R. So you notice there's a lot going on now in the console, very busy, but hopefully it should look familiar This is the first 6 lines of the data set. And this is the same data set we've been working with in Excel. You see some of the same column headings. You see some of the same row information for the specimens. And all of these information, histosex, dilutions that we've already seen. But one thing, when you do the head function, it gives you just a little preview. But let's say you're actually interested in looking at more of the information. The view, and make sure you capitalize the V there, the view function, which will run, will spawn this separate table. So now we've got a new window where we can actually scroll through the data set, look at all the information. Up and down, so columns and rows, right? So this is a way to basically look and see what have we imported. And so far it looks good. That's exactly what we want to have in. And up in the editor, I've got this one line here called fix. Let's say while you were viewing that table, you saw an error that you just wanted to quickly fix. You can use that fix command to do it, but our data looks good, so I'm not gonna make that change. And so at this point now, we have our data in, we've set a working directory, and we can begin with our analyses. And the format for this is gonna cover a similar approach to what Ken did. We're gonna have an introduction for each of these analyses, how to actually do the analyses, and then a plot from the output, and we'll have any questions on each of these sections. And we've covered the working directory and how to at least add and view data. So our first analysis is this weight-length relationship. And Ken's covered the underlying mathematical formula that we use to define it. And then the log transformed version of that, which is used to actually estimate those A and B parameters for our model. And so if we go over to our R window, we'll see that we can accomplish basically in this first line, a linear model of the log transformed length and weight. How we're doing that is there's this LM function that stands for linear model. And so we can now take that function, which is everything in the parentheses, and I guess I should say, you know, the standard, if you haven't already kind of noticed it, the standard structure is you have a function name, some parentheses, start and end, and then inside those parentheses, some attributes that basically then inform this function name and gives them some input for that function to work with. And so this is saying, let's create a linear model with log transformed weight data against log transformed length data using a data set called DAT, which is what we imported. That was our data table. And do all of that and assign it to this variable called WLR. And so let me go on that line and Control R to run it. So we look again down in our console, and that's executed. So let's now see what we have. So I've just now gone back in the editor. I've run this summary function, and summary tells us about the output of a model that we've run. And so here's the summary, and then it tells you what was the actual model that you're working with. This first line, this is the model. The next set, it tells you something about the residuals for that model, and then the coefficients of that model, as well as the fit of that model. And you'll see the R square down there, 0.98, this adjusted R square. So it gives you an idea of how well, with those parameters, your model fits your data. So let's move down. We're back up in the editor, move down to this command called coef, or coefficients. And so this allows us to examine the model parameters, or the coefficients as they're called here. So again, if I Control R, you'll see now in the console that its output, our intercept and our our log 10 length value. And that's probably looking familiar. This is the same information that we worked with in the Excel spreadsheet. And as we move down in our editor, let's select both the A and the B and Control R, run those. And so this now is our A, right? So this should look familiar. This is our A value, and this is our B value. And it's the same values that were output from the Excel spreadsheet. One thing to note, another kind of tip or trick here, is if you put an external set of parentheses around a line of code that is assigning something to a variable, you will both assign that to the variable and it will print to the console so you can see what the output is. And so that's why there's those external parentheses. And so that is we've just created a weight length regression. We've estimated the parameters, and now let's use those parameters to plot what that model looks like. So we're plotting this model. The first step we're going to do is create a range of X values. And so we've introduced another new function, seek. This is sequence. And so the sequence is a number that's from a starting number to an end number by some number increment. And so our seek here is we want the minimum of the values out of, we look in the data file in the column length, and that $sign means look in the column length and get the minimum value. So that's our from data, or our from value. And then with the max from that same column, that's our to. So we're going from and to, the min and max, and by 0.01. Okay, and that's our X value. And then for the Y values are based on our model, right? So we've estimated an A parameter, we've estimated a B parameter, and we've just created a set of X values above that we're going to call back to, and that then will create our Y values for our model. Those are both going to be input into our plot. And so this plot function allows us to take the length value and the weight value. These are going to be the points. And here's some other details about the plot. I won't go into them. It's just what are the limits, what type of icon, and then the labels in your plot. The next function, lines, is the actual model. So it's saying we're going to draw a line using that X data from the model and the Y data from the model. And then just some more details about the attribute of what that'll look like. And so let's hold down the Shift key, use the up arrow, select all of this, and then Control R and run it. And so now we've got the model output. And so this is the same plot, similar plot that we've seen in Excel. We're fitting that model to the data that we've observed using the best parameters that we've estimated. And we can also go over here and say, okay, that's a good looking plot. Let's save that plot. And dev copy and dev off are just, you turn this function device on and then turn it off, and it allows you to create a plot. And we'll look at that in a bit. So let's move to, how about questions on this weight length regression? Let's see. Is there anything?
Can you make that a little bigger?
Yeah, which, the text
so
or?
Your box where you're showing the R data on the left-hand side, if you could drag that out some more.
So is the font size too small?
I don't know, there was just some requests to make it bigger.
Okay. So wider or easier to read, did it say?
Sorry, I think that has been answered. I told her to actually click the and enter full screen. So she said it's already clear now.
Oh, it's good. Okay. So any questions? I'm looking at the Q&A. How to download the script from La Lima. So the script is in the Resources tab in La Lima. You go to Resources, scroll to the bottom, and select the stats in R. And it should be in that list. Okay, I see someone has a raised hand. Can know how we work for questions here, Annie.
Yeah, we have one person. So silahkan Pak Denny Suhernawan dari Universitas
Thank you,
Udayana.
Ibu. I would like to ask, when we already get the picture, what information we can get from the picture apart from the relationship between weight and length? Is there any other important information from the figure? Thank you.
Thank you for the question. The figure is just a visualization of the data and model. So there's not necessarily any additional information. Sometimes if you have a report or a publication, you'll include a figure, but it's really just to visualize. I will say often you want to plot the data as a way to identify outliers amongst your observations. That's quite a useful tool for plotting. But typically for something like a weight length model, just reporting the equation with the A and B parameters is sufficient for most cases.
Okay, thank you, Mr. Eric, for the explanation.
You're
Terima
welcome.
kasih, Ibu.
Sama-sama Pak Denny. Ada pertanyaan lain? I haven't seen any raised hand yet, actually. Pertanyaan lain, Bapak Ibu? So I guess if you don't have any question, you can continue with bimodal size distribution, Eric.
Okay, thanks, Annie. So moving on. So as Ken has covered for us, the sex-based bimodal size distribution is a way for you to compare male and female lengths and their distributions. And we've had some comments that I just want to bring up the fact that what we're presenting is because of the time and the diversity of the audience, it's a very simplified approach to this problem. And there are some additional, more detailed steps that I think are useful or needed, but often if you don't have a statistical training, it's challenging to understand how to interpret and do them. But one of the questions that we had just that I'd like to cover is this assessment of normality as a first step. And some of the things that you would do for that are you would visualize your data in histograms. You would create QQ plots. And I'm not going to go into details of what these mean, but if folks have some experience with them, you'll know what I'm saying. And then you can test them using a Shapiro-Wilks, or there's a number of other tests for normality of your data. That can be an initial step. But for at least what we're presenting, we're somewhat assuming you've got normal data, and then moving on to that test for the homogeneity of variances, or that F test for that unequal variances. And so we'll go into some of those details here and how to do that in R. And then also having a plot, how to visualize this. And so let's scoot back over into the editor, and I'll scroll down to the bimodal size distribution. And one of the first steps that we want to do is subset our data so that we can now work with just male data and just female data. And there's another note here that I want to bring up. The use of brackets after a data set, or data frame as they're called in R, allows you to subset those data sets. And as I mentioned earlier, this $sign identifies a column that you might want to pull information out of from a particular data set. And then when you're using subsetting, a double equal sign means it's equal to. So it's basically checking equivalency. And then this just straight up and down vertical line represents an or. And you'll see why I'm going over these. So if we want to subset our male data, we're going to call this a new variable called dat.m. And to create dat.m, we're going to use dat. So that's that original data set that we brought in. And we're going to subset it, right? So here's a bracket, and there's the closing bracket. And within that bracket, it's saying, look, it's this first set of information in front of the comma represents what you do with the rows. And then the next is what you do with the columns. And so it's saying within this dat, we want to look for any of the records or rows that for the histosex column equal the term MIMM or male immature. And then this vertical line means an or again from the histosex column, any of the records or rows that are MMAT or male mature. And then we want to select those, and we want to bring all of the columns with those rows. And we want to then assign that to this dat.m. The next thing we want to do is create a new variable called sex. And to create that, we're going to use a function called rep or repeat. And so all it's doing is repeating the letter M that represents male. And it's going to do that a number of times that are the same as the length. So basically this is a count of how many records are in this histosex column that's found within the dat.m data set that we've created in this prior line. So repeat M this number of times and assign that to this new variable sex. And then dat.m, we're actually going to overwrite that first dat.m by cbind, means bind two columns together. And the columns we're going to bind are that original dat.m that we made up here with this sex variable that we just made here. And so let me select all these three, Control R to run them. We see they're populated down in the console. And so we've now just created this new data set, dat.m, so for males. And using the exact same syntax, we're going to do the same thing for females, but just call it dat.f. So instead of go through that, I'm just going to select them, Control R to run them. So now we have male and female data sets. And again, we're assuming here that we're just working forward and moving forward with normal data. And so we're going to combine that sex variable. So we've got male and female sex from our dat.m and dat.f. We're going to now R bind them. So the C bind was taking columns and bringing them together. R bind is taking rows and bringing them together. So we're going to R bind the dat.m and dat.f. So that means we're basically putting them together to be a single data set, again, called dat.sex. And now let's populate those, or sorry, not populate, select, and I'm going to run it. And so this should look familiar. Some differences, right? If we look at the specimen column, we notice it's not in order anymore, because what we've got now are all the males in histosex that are at the top of our data set. And we've also created, if we scroll all the way to the end, this new variable called sex. And that will be used in an analysis coming up. And then if we scroll down, we see that we have Fs. So these are the females down here that have been that have been concatenated up into the data set. So this is our new data set we're working with. So I'm going to close that. And so the F test in Excel is called a var test, VAR. And so this will run. So I'm selecting it, Control R, and we see we've got some output. And again, the output to us suggests that we've got unequal variances. And this next set of information, I've commented all of it out. But if you did have equal variances, you could run a t-test. And so this is the exact same test you're running in Excel, but in R. And it's pulling the DAT-M and DAT-F data, and it's comparing these two. Okay, it's saying, is the length information from the male data different than the length information from the female data? And in this case, you see this attribute here, var.equal. So that means variances are equal. If they were true, that's what it would look like. But since we know it's false, we're going to run this version where var.equals equal false. So let's run that, select it, Control R. We see some output now has come into the console. And from the results, we found that there is a statistically significant difference between these distributions. And in the case of the t-test, it's testing against the means for these distributions. And so let me scroll down a little bit. So we've got the model output. Now, how do we create this? How do we create this figure that we've shown? So we're going to plot two histograms, one for males and one for females. And there's a lot of detail here, but part of what we're doing is just introducing some descriptive details to the attributes of the plot so that it will look pretty when we plot it out. So this S variable is just a measure of, it's basically saying we want to identify the minimum values that are both in the male and female lengths and just give a little bit of an offset just to move it over a little bit so we make sure everything's included in the final plot. And then L, we want to take the maximum value of all the males there and all the females. And so it's basically the maximum value out of either male or female. And assigning that, smallest is S and largest is L, okay? And then we're creating this AX variable for the axes. This helps us with the breaks. And so a histogram has bins, and this defines our bins. And this pretty function is a function within R that rounds off values to 1, 2, 5, or bases of 10 that make for prettier plots so you don't have odd numbers on your axis. So I'm going to select all these. I'm going to Control R to run them. You see they've executed in the console. And then the histograms, this function hist, this is create histograms. But what I'm going to do is actually just create the histogram data for both the females first and then the males using the breaks as defined by this AX variable that remember is, oops, didn't mean to do that, that remember we created with this pretty function. And that's just rounding the bins to something that is visually pleasing. And we have this option to say plot false, meaning create a histogram, but don't actually plot it out, just hold the data. And we're going to assign that to HG for histogram F for females and HG histogram M for males. So let's select and run that. And then we need some colors. So the C1 and C2 are going to be the female and male colors, and they're based on this function RGB. And so R is red, G is green, B is blue. So that first is the R. So if you see 255, you know that's red. This second one is 255 in the third, so that's blue. And the alpha, max is just the intensity, but alpha attribute tells you how much transparency there is. And so this is a way that you can overlap your histograms within R. And if you had a 0 transparency, then it wouldn't plot because it would be an invisible plot. So let's define that C1 and C2. We select, we Control R to run them. I'm going to scroll down a little bit here. And so to plot our histograms, we're going to plot 2 histograms, right? The first one is our female histogram. The second is our male histogram. As you can see right here, this is the object it's plotting, hg.f. That was our female histogram. It's taking that data and it's using color, COL's color, defining that as color 1, which we just created up here. And then YLim is just the height limit. The Y limit goes from 0 to 55. There's no main title. And we've got an X label and a Y label. And then we've got this second plot for the male histogram. And we're going to add that. So the add true adds it to the first plot. We're doing a few other things here. We're going to add what are called ab lines. And if you see a V, that means it's going to be just a vertical line. So we're taking the mean of the female lengths for the first line and the mean of the male lengths for the second line. And we're going to plot those on top. And then the legend just allows a little box in the top left-hand corner of the plot that will list male and female and what colors they are. Okay, so let's select all this and run it. And somewhere hiding here. So it's not, if you run a plot and you don't see it, sometimes you can click the Windows drop-down menu. You see graphic device. That's where it was. It was just behind all of our code, our console and editor. And so now we've just created this figure that's the same as in the talk. And it presents in red. This is the histogram of the length frequencies for the females, and then in blue, the histogram of the males, and that purple, right, is the overlap. So you're seeing the females kind of overlapping towards the males, and then that long tail of the males in purple. So it's a way to visualize both. And the means are the vertical dashed lines for each sex, color-coded. And again, if we like that, we can save that plot, select it and click Control R, and that's written into our working directory. And so that's how you do the sex-based bimodal size distribution. Does anyone have questions on those methods? I think I see some.
Yeah, we have one person, Ibu Husna. Ibu Husna, silakan bertanya.
Yeah, excuse me. I already installed for the R. However, I couldn't find the R editor. I only find the R console. So how to find R editor?
So what you want to do, have you downloaded the stats in R file?
Yeah. Yeah.
Okay. So if you have the stats in R file, you can click this open.
Yeah.
And then navigate on your computer to where it is. And it's in the training folder for me. Click that. And if you click open, I'm just going to click cancel because it's already open. You'll see this window open up, and it should have the editor with that script inside it.
Okay, thank you.
You're welcome.
And second question, Pak Denny. Pak Denny Suharnawan, silakan Pak.
Ya, terima kasih Ibu. Oke, brief question. Could you please repeat the meaning of the purple? I mean, red and blue, and what is the meaning of the purple? Okay, thank you.
Yeah. So the purple it's a transparency. So let me demonstrate. So if we go back here, and we put alpha to 0.
Yep, alpha to 0.
And then I select and rerun everything, and then view the window. So do you see the red is the distribution of just the females.
Female.
And then the blue, I'm going to close this now and let me rerun again.
Yep.
So the blue is the distribution frequency of the males. And the purple is I've turned on a transparency for the males so that we can at the same time see the distribution for the females and the males. And so the purple is where there is overlap in both the males and the female distributions.
Okay. Clearly, very clear. Thank you,
Okay.
Professor.
You're welcome.
Terima kasih.
Terima kasih, Ibu.
Sama-sama. There is a question in the Q&A box, Eric.
Okay. I don't hear, help me, the other one.
The other one.
Explain the R analysis based on statistical analysis file.
Yeah, he was asking a PDF file.
PDF. Okay, so this statistical analysis, that file is just the PowerPoint. So this is this file up in the upper right-hand corner that has all of the slides that I'm going through. So that's all that statistical analysis in R file is. It's a PDF that represents the PowerPoint.
And I think the PowerPoint will be shared in the Laulima portal, right?
It is in there now, yeah.
Okay, great. So, ada lagi pertanyaan Bapak Ibu? I think we don't have any more raising hands for
Great.
now. And now we are on a break for 5 minutes. So, Bapak Ibu, sekarang kita break dulu 5 menit ya, sebelum nanti dilanjutkan oleh Pak Erik menjelaskan tentang size at maturity. Terima kasih, sampai ketemu lagi 5 menit. 5 minutes.
Spawning stock biomass. And a typical plot of that will look like something like this. And there's the general mathematical equation, how to calculate it. So let's jump back to R. Looking, yeah, okay, it looks good. So we're going to start with female size at maturity, excuse me, and using the length bin data. We're going to start by assigning or creating a new variable and assigning it a number. So this bin width is basically going to be based on centimeter length bins. So we're saying we want 2 centimeter bins, and we're going to create 2 variables here to help us determine those bins. One is the size min and the size max. And to calculate the size min, we're going to take seq, remember that's sequence, so from, and it's the range of values, but it's when you have this bracket here again, remember that selects out of a data set. And so the range is giving us 2 numbers. And the first number in the range from the female lengths is the minimum size. And then the 2 number, this is the maximum size that we're going to find. So I'm sharing different ways, different functions for you to be able to get these kind of information out of data sets. And then by is by the bin width that we've just described in the prior line. And the size max is the size min plus the bin width. And this is similar to the analysis preparation that's done in the Excel spreadsheet where you're creating your bins for the size and maturity. So hopefully, even if the code doesn't look familiar, the process will feel familiar. And then you've got these 2 sets of values that you now call the bins variable, and we're sorting them so they're ordered, and it's just the numbers for the size max and size mins. So we are now going to select and run all of those. And the next step we need to do is calculate what's the average length in the bins. So here's a new function, this sapply, and it's basically a very powerful looping function within R. That's one of the base commands, and there's different kinds of apply functions, but this sapply, I won't go into too much detail on them. They're very powerful and useful if you work with R, you're familiar with them. The sapply will identify across this number of values. So length is basically a count. So it's a count of how many values are in that, saying 1 to that number. We're going to execute a function, and that function is taking the mean of these values. I'll scroll over a little bit so we can see what's going on. It's the mean. Oh gosh, where'd I go? A little too high there. Sorry, here we go. So it's the mean of these values that are any values greater than or equal to that size min and less than or equal to that size max. And we're identifying that they're the lengths. And so this I is a counter in the sapply loop. And so the things that you want to count through, it's going to assign a number, and it's sequential, 1, 2, 3, 4, for as many values as there are from 1 to this number that we've calculated from the length. And the reason why we don't have a particular number is because if that changes with the input data, we want that code to be flexible enough to change with it. And so we see an I here, that's the counter, an I here, and an I here. So that means it's going to cycle through those values and then give us a vector, which we've created here to hold the data, that's the average sizes. So let's select all those, let's run them. And in a similar way, we're going to now just count the number of females in each bin. So n.f, number of females. And we do something similar. So the sapply is a kind of a looping function. You can also explicitly write a for loop. So this is saying for I in, and again, it's the 1 to whatever this number is, execute this. So take a sum of all of the counts that are either greater than the min or less than the max, and then sum them up. And in each value, as you loop through that I counter, assign that to this n.f, or number of females vector. And so we're doing that for each bin, basically. And so now we're going to select Control R, run that. We do the same thing for not only the number of females, but the number of mature females. So I'm just going to select, run that again. And this is probably looking familiar. This is the same kind of table that you created in Excel, but you're just creating a set of vectors that hold that information in R. So freqmat, frequency mature, we're now just taking that number of mature females, dividing it by the number mature in each bin, and assigning it to this frequency variable, this freqmat, frequency mature. So let's select and run those. And now we finally get to the point where we're actually going to run the size of maturity model. And so in R, or I should say in Excel, you use solver, you had the actual structure of the model, and you input some information. In R, you can use this GLM function, which stands for generalized linear model. And you can assign to the family attribute, I've got quasi-binomial. You could also do binomial. We have some values that are not just integer values, and so the quasi-binomial will handle the non-integer values. And this is just basically saying, I want to run a logistic regression on our average size data and our frequency mature. So this is the same information that we used in Excel to calculate that size at maturity. So let's select all that and run it. And as we've seen, the console's being populated. And now we can actually calculate, excuse me, our L50 from a coefficient that came out of that SAM model, the size at maturity model. It's actually the A and the B, a negative A coefficient divided by the B coefficient. That will give you that L50 value. That's a standard equation there. And we'll also calculate the L95 for input to help us plot our model. So select it and then Control R again to run. And we can look down here and in the console now, we've got output, and the intercept in this case represents our L50 value, 23.9. And you'll notice it's not exactly what is generated by the Excel estimate. It's quite close, but part of that has to do with the estimation procedures that are done in Excel and R are a little bit different, but it's still close enough to be biologically similar. Okay, so that's at least the statistical output. Now let's plot this output. So similarly to what we did for prior models, we're going to create some just dummy X values. And these represent a range of lengths using that sequence function. And then we're going to create the model predicted values using this predict function. Predict is powerful. It allows you to take a model object. So again, we used the GLM function up here to create a model object and assigned it to this SAM-F value. We can use that same SAM-F as input to predict and say, okay, use this model and the list of inputs that you want for the size average. See, this is the model. Remember this size average. Those values are going to be provided by this range of the x.sam.f. So this range, the sequence of values that we created over here. And the response is just basically the probabilistic response. So this is the logistic response that we're putting in there. And then the plot, so these are the X and Y data that's going to help us with the model plot. The plot, here's the observations, right? Here's the observational data. So it's going to be the average sizes and the frequency mature at those average sizes. And we're going to have some dots and we have some labels in here as we've done before. And on top of that plot, we're going to create a line. This is the model. So this is the model that's fit to the data. And then we're adding a couple, a vertical line. So the AB line, remember, and this is V. So it's saying we're going to create a vertical line that is the same value as the L50. And then a horizontal line at this 0.5, which represents that half the mature proportion. And then just some text to help identify the actual value of L50 on the plot. So select all that, Control R, and this is the plot we get. So in the crosshairs there, right? This is where our L50 exists. And how do we find that? We come over, we look at our proportion mature, 0.5, that's 50%. That comes over where that intersects our curve, our model. Then you go down and you find, oh, what length is that at? And that length here is 23.95 centimeters. So that is our L50, and that's how we've plotted it. And the dots are the observed values, and the line is the model fit data. So are there any questions about the analysis for the size at maturity and the plotting of it? Let's see, lots of Q&As, yeah? No?
There hasn't been any. Okay, we have Ibu Lilis. Ya, silakan Bu Lilis.
Thank you, Ibu Eni, and thank you, Professor Eric, for the presentation. I'd like to ask about the reason of using the GLM in this case, yeah. So why did you use GLM instead of LM? perhaps just for your explanation. And also later on, do we need to consider like to use the the checking of the assumption of the GLM itself? I'm not sure, but I think in the, for example, in GAM additive model, we use GAM.check, GAM check. Perhaps in GLM we use just plot the GLM. I'm not sure. So do we need to consider the results of the checking of the assumptions, or the like not including the QQ plots for the normality test? Thank you.
Thank you, Bulilis. Good to hear from you. So to address your first question, so the GLM function, I use it instead of the linear model function because this allows me to have a link function that can define a logistic regression. And if we look at the plot, even though, interestingly, the data that we have actually would probably fit a linear model fairly well, often you will have size at maturity data that doesn't look this good and is more sinusoidal, so S-shaped, which is the standard kind of function that you'll use to fit these things, because you're moving through this size classes where you've got almost a little bit of maturity all the way to full maturity. And the more they shift so that this line in the middle becomes vertical, that almost becomes a knife edge selection. But the GLM here is a function that actually allows you to do that sinusoidal curve, whereas the linear model is really just, it's fitting just a linear model. It's an ordinary linear regression. So there's much more flexibility in the type of model, in this case, the family attribute that you can define. So it can be binomial for logistic regression. So that's why we're using that. The other question about the linear model assumptions, we're not testing for those here. They're not necessary for this. I think the difference between the prior tests where we were looking at comparing normality and some of the underlying assumptions about the structure related to more the t-test approaches also. So in those, because we were looking at comparing variances, the f-test that we use for that actually requires that the data be normal. And if it's not, that's when you would then potentially use a non-parametric approach, something like a Wilcoxon ranks test, as a way to compare between your populations. And so those I think those are some of the methods that you would use there. But as with any regression, you can also look at the residuals of this model to look at the performance if there's anything out of the ordinary. But the data for the size of maturity is quite a bit different than fitting, say, some of the other regression model approaches that we've looked at. So I hope that answers everything, Lilis.
Yeah, thank you, Prof. Eric.
Okay, you're welcome. Terima kasih.
Okay, Eric, we have two questions in the Q&A with regards to the R script. Perhaps
okay.
Have a
yeah,
look.
so first question, I think the script is a formula and the R is the machine. So when you can connect your own data CSV with the original template, it's easy to create this analysis and edit whatever you want with the access level or anything. Thank you. Yes, that is absolutely accurate. And at the end, I will show you how you can do that. And it's very straightforward. And then the second question, we can also calculate L50 using packaged Tropfish R based on length weight. How close the result if we compare with histology data? So I have looked at Tropfish R. I can't remember if it actually uses, if you input data to calculate L50s or if you're using, say, family-based models. So for instance, if you work with snappers, if it's using an assumption about the longevity of snappers and the max size in your population and where the L50 of your species may be. I don't know if it's doing that or if it's actually calculating a size of maturity using input. If it's calculating a size of maturity using the input data, you should get very similar or the same results as what we're presenting here.
Okay, so we don't have any other people raising their hand. So I think you can continue on sex ratios, Eric.
Okay. Sex ratio, next up. So as Ken presented earlier, there are a few different steps here to looking at sex ratios. Oh, and just for folks, I have actually included a means to calculate the male size of maturity, but we won't cover that right now, but it's in the R code for you to work with. So sex ratios, next up is the overall sex ratio. And so this is for all the fish, all the males and females, not just the mature fish, which is considered the operational sex ratio. And so again, we have another note here that I've included in the code about the grep or grep, I've heard people say it both ways, function. And this is a regular expression function that provides a text match from your data frame. And again, that length function counts how many numbers of matches. And this caret, so this little upside down V, before a letter, in our case, F or M, means we want to find text that starts with a capital F or a capital M and identify that. And so what we're doing, we're using this grep to find words that start with capital F out of the dat.f data frame in the histosex column. So as you recall, we classified things as either FIMM or FMAT, whether they're a mature or an immature female. And we've done the same for males. And so this is just a handy regular expression, excuse me, to allow you to find any of those that are either immature or mature and pull them out. And then it will, with the length, count the number of those occurrences and assign that to this observation. So this is the number of females observed and the number of males observed. So let's select and Control R. And then we're going to need to get just the total number of observations. And that's quite straightforward. It's just the female plus the male observations. And then we want an expected value of female and male observations. And so generally in biological populations, you would expect there to be a 1-to-1 male to female ratio when it's sexually reproducing populations. And so our expected values here, what you can do in R is if you have two variables that you're creating that are the same value, you can actually kind of link them together, as you see with this expected F equals expected M equals, and then this is the actual value that you can assign to those. So I'm going to select all these and run them. And let's scroll down a bit in the editor. So the next step is we're going to actually calculate the overall sex ratio. And the overall sex ratio is really only this part in here, this observed males divided by observed females. But what I've got around here is I've assigned it to a variable called ov.sr, or overall sex ratio. And we've got a new function to introduce, the paste function. So the paste function allows you to paste together different pieces. It can be text like we have here. So this is just going to be exactly what it says. Overall sex ratio is, and then comma, and you can now paste that together with round is a function that allows you to round a number, and the number is going to be calculated by this observed.m divided by observed.f to 2 digits. And then also comma, paste this last part, :1. And we'll see what that looks like. So let's select that. And we run it. Actually, what did I do? Oh, because I assigned it to a variable. Well, let's do this and you can see what it looks like. So I just went down into the console, and since we've assigned all this to this ov.sr, when you actually just type that in, it's going to print out what is held in OVSR. And it's this overall sex ratio is 1.08 to 1. And if we look back over here, these are equivalent. Let's see. Covering up. So this 1 to 0.92 is equivalent to 1.8 to 1. They're exactly the same. It's just whether you want the 1 to be represented by the males or the 1 to be represented by the females. So that's equivalent. And that's what we've done in that function. And then we're going to calculate the probabilities of the overall sex ratio. And that's just the females. So it's the females divided by the total and the males divided by the total. And these are going to be all inputs into a contingency table that we'll use to statistically test these results. And so here's our expected value. Our probabilities are our observed values. And now we get down to the test. This is the same test that, that Ken has run in Excel. This is a chi-square, and we're looking for our output, right? We want to see what's the p-value. And in this case, p-value is above 0.05. So what does that mean? That means there's no difference from a 1-to-1 sex ratio. So that 1.08 to 1 is basically statistically the same as a 1-to-1 sex ratio. So moving on to the operational sex ratio. And again, this is very similar syntax, so I'm not going to go over it. But all we've done is added this M at the end. So it's very similar to the others, but not the same variables. And that just means female mature and male mature. So I'm going to run these, select and then run. Do the same thing for our observed and expected proportions. And here we're going to calculate our operational sex ratio, but hold it in that variable. And select again the observed and expected values and running those. And finally, we get back down to our chi-square contingency test again. It seems to have disappeared. No, where is that? Okay, here we go. Okay, and now we look at the output. And so what's our p-value? Our p-value now is 0.02. And if we look in our notes there, if we've got a p-value less than 0.05, it means that the sex ratio is different than 1-to-1. And so if we move our notes ahead here, the p-value is 1.31 to 1 or 1 to 0.76. Those are mathematically equivalent. And statistically, they're different. So what have we found? We found that there's actually the sex ratio, the operational sex ratio is skewed towards males. And so we like those results. And we're going to actually now create an output table. I'm not going to go into all the details here of this text for the output table, but the sync function allows you to basically write to a table that's outside of R, inside the our working directory. The cat is just, think of it as a line that goes into this text file we're making. And so the first is you can say cat and do OVSR. So I'm saying in that first line, I want you to put in that overall sex ratio output. In the next, and then we've got a semicolon. That means we can execute both of these functions in the same line in the console. And this, within parens, this \n means new line. So it's very literal. So you're writing everything out. In this next line, we're pasting together the word method and then the OVSR test method. And just, that's basically just going to say chi-square test. And so all of this information now is being written out to a sex ratio text file. And I'll just select it. And when you use sync, you need to have a start and an end function for the sync so that it closes. So Control R, and we've just written, and I'll show in a little bit how we do that. Or sorry, I'll show in a little bit all these files that we've been creating. And the last sex ratio that we're looking at is this size-specific sex ratio. And so for the size-specific sex ratio, these are very similar approaches to what we did for the size at maturity, where we're creating a size min and max for the bins. So I'm going to run that. We're calculating the average size for each bin. We're counting the number in each bin. We're counting, sorry, the number of females in each bin was the last one. This is the number of males in each bin. Now we're getting a total from the counts of females and males. And since we're again doing a test here, we're going to calculate our expected numbers. So the expected females equal the expected males, this 1-to-1 ratio, or 50% of each. And now we're again calculating female, male frequencies and what their ratio is together and running all those. And now, which is a little bit different than the operational overall, we're actually going to generate in this chi.p variable a vector of chi-square values that have been generated by a loop. And so instead of just saying we have one chi-square test, we can actually run this chi-square test across all of the information for each size bin and then have them write out the value and use that to evaluate whether or not they're significantly different from a 1-to-1. So let's select all those and we'll run them. And so what's interesting here is we've got some warning messages. So what's going on with the warning messages is that there are some zero values, which Ken had, I think, pointed out earlier, that chi-square estimations don't like those zero values. So the test can still function, but it's just letting you know that it's going to make an estimate that you need to check. But in our case, it's fine. It's just a warning. It hasn't broken anything. And then what we can do, we've used all those functions to basically create all this output and the size-specific sex ratio. So we're going to create a new data frame and call it this SSSR and take the minimum size data, the maximum size data, the average size data, the counts, the female counts, the male counts, the expected values for the females and males, their ratios, and the chi-square values. So let's create that and let's view it now. So this table should look familiar to what was in in Excel, where you've got size min class. So these are all your different size classes, the min and the max, and what the average size is in there. How many fish total are there? How many female fish there are? How many male fish? What's the expected numbers? And then what are the ratios? And what are those chi-square values for those different ratios? And remember those errors that we had on the chi-square, that's for those three last values where you've got an infinite ratio, and that's because there are no females there. But we can still evaluate the fact that that it's only males. And so there's definitely a different sex ratio for those size classes. So let's close that table. And let's visualize those results. So we're going to use our plot function and look in this SSSR data table, data frame we just created, and pull out the sizes, the average sizes. And then we've already got this frequency of females that's already outside there. So we'll use those two as the X and Y data. And then with some details about what the plot will look like. And then to create the lines, in this case, unlike what Ken did, I'm actually just going to connect the dots, literally, with lines. And so it's the same information as what goes into the plot. And we've got one ab line that's an H, so it's horizontal at that 0.5 or 50%, 50% line, just to show you what that expected 1-to-1 ratio should look like. So let's select that, Control R. And again, we don't see it. Let's go up to our window. And there's a graphic device. So let's select that. And here's our size specific sex ratio plotted out. And again, this isn't a model that's fit to it. This is just the data plotted with that 0.5 or 50% proportion female to give you an idea of where the 1-to-1 line exists. So that's everything for the sex ratios. Does anybody have any questions about those?
Yeah, there is, okay, Pak Denny lagi. Silakan Pak Denny.
Okay. When we find a data, no female or no male at a given size, is there any the meaning when we find out such the data? Say in a given size, only male, not female, or vice versa. Thank you.
So that's an excellent question. And I would say that is an area of active research, and especially among our group, we're very curious about what may be driving these size-specific sex ratios, especially for species that don't sex change. And so we see some of these species that, we've had different ideas. Is it, if you only see large males, is it because there's differential mortality where the females are experiencing a higher level of mortality? Is there a senescence where they're actually dying earlier as a result possibly of the energy that it takes to create eggs for reproduction? So the answer is I think there's different explanations that could be there, but it's fairly wide open. I mean, I'm open to hearing if you have thoughts on why you might observe some of these. It could be, in some cases, is there a gear bias? Is for some reason it's only targeting, the sexes are in the wild separate, and the gears are for certain sizes only targeting one sex. That seems not as realistic as the fact that they're just not there. But I don't know. Do you have any ideas of why you may observe this phenomena?
Yeah, it means that the data is still open. We can explain to maybe related to mortality. Also, can we connect the data, say in a high length, like in the graph 35? Can we connect it to that? Maybe there is a sex changes. From female to male or vice versa. Is it possible?
Absolutely. So you could see this sort of graph for a sex changing species, and you would expect that to be more common as an explanation for why you would see this. But we have seen this for fishes that do not change sex. And so that is the interesting result of why you wouldn't have that 1-to-1 throughout the whole size range.
Oh yeah, it's wonderful data. What about, so, okay, could I another, what about in minimum size in length? Example, maybe less than 20. So when we don't find a male or female, can you have any idea to me to explain about the conditions? Thank you.
And that, I mean, it's the same explanation, but imagine this graph, you know, tilted the other way, where you've only got all males that are the smaller fish and the females, you know, observe. It could be, perhaps they're not, at least if you're using histological based approaches, maybe that size class is not identifiable yet as a female. That seems unlikely.
Yeah,
it could be a sampling issue, but we do see all different combinations of this, where it's a lot of, you know, more females are bigger and there's no males. We see all big males and no females. It's a little harder on the smaller end, only because with immature fishes, identification is problematic, and so often you don't actually know what sex those smaller specimens are.
Oh, I see. Thank you, very clear explanation, Professor. Thank you, Bu Ani. Kami kembalikan waktu.
Sama-sama. We have Ibu Regina. Ibu Regina, silakan Ibu. Ibu Regina, mau bertanya, Bu? Okay, we don't hear from her. We have a question, Eric, in the Q&A from Fabian.
I see that. Okay, so Fabian, the question or comment is in L50 error in coefficient sam.f, object sam.f not found. So if you have that kind of an error, it probably means that you have yet to run the code. So the line of code that will create the model, that the size of maturity model that will then assign that output to the sam.f variable. If you haven't done that yet, you're going to get an error later on where it hasn't been created yet. So you just need to make sure that you go back and run that sam.f line, and then you'll have that and can use it.
Okay, I think we don't have any more that ask question, so we can go to fecundity, Eric.
Okay. So fecundity, for fishes, a fairly standard assumption is that as fishes grow, their potential reproductive output or egg output grows exponentially. And so this model is the same structure as the weight length model in terms of the mathematical equation that's used to define it. So if we pop back over to R in our editor and we start in the very first, this should look familiar, this LM. So we've got a linear model again, and we've now taken the log transformed fecundity and the log transformed lengths from our original DAT file. And instead of, this is very similar to the weight length, but instead of weight, we've got fecundity, and I'm calling it batch F or batch fecundity. So let's select and Control R to run it. And so now we've already run our model. We've got coefficients or parameters that are sitting, waiting for us to look at. So let's do that. Let's select the next set and then Control R. And if we come down and look, we've got A is this first value, the 6.36 to the -8. And then our B value is 8.3. So that's quite a bit higher than the typical 3 that you would expect from the weight length relationship, but it's purely valid. It's valid for the data that we've presented here. So that's the model and the model output. And now similar to the steps that we did for the weight length model, we can plot the fecundity model. So we create a set of X values, and I'll just run that. and then we walk down, and we now will create a set of Y values, again, from our model using the, yeah, we don't need that, using the equation for our model and the parameters that we've estimated. So I will just Control R, run that. So now we've got those two sets of information, and we want to do a plot of the observed data points. So those points are the lengths and the fecundity from the DAT file. And again, these are just details on the plot, what kind of points we use and the labels we use. And then we're going to write a line over the top. We're going to draw a line that is the X and Y values from the model. So let me just select those two, Control R to run it. And so here is a graphical representation of our batch fecundity at size model. And if you like that, you can then go back over here, select Control R. And again, we've just written this plot out to a PDF in case we need to use it for a report. So any questions about this?
We have one person who raised hand, but she never answered. So
Okay.
In that case, we don't have any other people that ask question about this.
Yeah, this is not that different from the weight length modeling. So it's really just changing some of the labels, fairly straightforward. So I'll continue on.
Sure. We have Pak Denny again asking question.
Okay.
If it's okay with you.
Yeah, absolutely.
Silakan Pak Denny.
Thank you. I'm wondering with the value of B, you said that in body weight length correlationship, maybe value of B more or less 3. In fecundity could be until 8. Is there any criterion when the value of the value of B? Thank you.
Yeah, thank you for the question. Logically, you would think it's probably similar to 3. We've got 8 here. This is, to be honest, this is simulated data. This isn't actually observed data. And so the value is quite high. It's just that the model is fitting it well. But if you've worked with fecundity data, you'll find that the data can be extremely noisy, meaning there's a wide spread. And often you'll find that it sometimes does not conform to any good model fit, meaning it's kind of that shotgun of values that just, you know, a zero slope linear model is the best fit, which essentially means there's no relationship with size. You may find at times that a linear model with a positive slope is also as good or a better fit than this model. But in terms of guidelines on the B value, you would theoretically think it should conform towards 3, but if it's greater than that or lesser than that, it's to be expected, because as again I said, this data can be extremely noisy, and at least you can see from our plot a lot of this is, you know, driven by this one kind of these couple data points that are quite high. So
yeah, it is because sorry.
I was going to say, if you're able to actually fit a model to fecundity data, consider that a success.
Right. Is it because fecundity is individually? I mean, different individual could be different. Is it? So the data is very noisy.
It's very noisy. Part of it's very noisy. Often you have a very low sample size, but the information also is very noisy, because imagine you start with a gonad, and then you cut a very small section out of that gonad, and then from that you liberate, you use some chemicals to get the eggs out of that very small section, and you count a sample of those eggs, and then you take those counts and turn around and extrapolate that number back to how many total eggs you think are in the gonad.
I
And so
see.
there's a lot of
Oh,
uncertainty, a lot of noise.
yep. Oke, oke. Thank you, Profesor. Terima kasih, Ibu. Waktu kami kembalikan.
Terima kasih. Okay, I guess now we are on to spawning periodicity, Eric.
Okay, great. Thank you, Annie. So spawning periodicity, as Ken has shown us in the prior session, is based on calculating a gonadosomatic index and time. And here time can be something related to an annual cycle or a seasonal cycle or monthly or on lunar phases. And our GSI is calculated with the gonad weight divided by the body weight, but with the gonad weight subtracted out, and then times 100. And that's that GSI index or statistic that we work with. And so if we come over to our R script in the spawning periodicity, the first step we want to do is calculate those GSIs. And so this first line, if we go back to the PDF, this lower formula is basically this line. I have the 100 at the start of it instead of the end of it, but it's basically the exact same formula where we're taking gonad weight, body weight, whole gonad weight subtracted out from body weight, and calculating the GSI. And so that's assigning it to this new GSI variable. And now we're going to again take the cbind function, column bind. So it takes columns and puts them together. So it's saying take the dat frame and put the columns, append the columns for the GSI to that and call it this new dat.g. And to calculate female GSIs, we're going to create this new dat.g.f, and the F here is for female. And so we've got the dat.g, and we're doing two things. We're using, again, the brackets. Remember, when we use brackets, it means we're subsetting within that data frame. And in front of the comma, everything has to do with how we're selecting rows. And when it's blank behind it, that means select all columns. We're going to include all columns. And so it's just saying, okay, within the brackets, from the data.g histosex column, for any records that are equal to either the F immature or the F mat, F mature, select those and assign those rows into this new dat.g.f. And then we've got one more step. This is the step that Ken has reminded us of. This is to make sure, because right now we've got all females. Remember, we've got the immature females too. We want to further subset to just select those fish that are at or above the L50. And I've got a little note here, and you can put the notes at the end. So you just have the hashtag and then you can comment. Make sure you calculate that L50 so that you can actually run that. And then what's the next step? We want to take the mean and the standard deviation of the GSIs. And remember earlier we used the sapply function, which is a looping function. Here we've got a similar type of function, a tapply function. And that allows us to look at, in this prior data frame that we just created, in the GSI column, and for the months, the unique, excuse me, the unique values in months that are in that table, we're going to calculate the means of the GSIs. And this NA.RM means if you have a missing value, remove it. So NA, remove, not available, remove. We want that true, because otherwise it will not allow us to calculate a mean. And we're going to do the exact same thing, same syntax, but instead of mean right here, we have SD. SD in R means standard deviation. And so let's run all of this stuff. And if we look down into our console, we'll see, excuse me, we'll see what looks very similar to the tables that Ken presented, where this is the GSI mean, and this is the GSI standard deviation value. And the 5 through 9 represent the months. And we'll clean that up a little bit. But let's say you don't want to look at it in the console. Remember this view function up here. So let me control select and control R. We can look at the same thing. Okay, so the means and the standard deviations. And we see that, for instance, the mean is highest in the 7th month of the year. So that's July. Okay, so we like that data. And what we're going to do now is actually create a table of it and write it out so that it's not just in R. We've got a new note here, and it's the semicolon allows you to run multiple commands on the same line. And so this semicolon here allows me to do two things on the same line. I can create this new GSI.f by binding together. So I'm going to call now the months are those row name values. Remember the 5, 6, 7, 8, 9? I'm going to now call those months instead of row names and bind that back to the original data set. And then that row names variable, so it's not redundant, I'm just going to get rid of it and call it null. And then what we'll do is we'll take that data set and write it out to a CSV file called GSI_F for GSI female.csv. And we're using this write.csv function. So that's saying, okay, you can write this data frame and call it this, and it's going to put it into our working directory. So let's select all those, Control R to run it. And so even though we haven't seen it, it's written out a table for us. So now we have a table that we say, but let's also do a plot. So our plot here, excuse me, our plot, we're going to grab the names of our GSI means. So this is the months. And then we're going to grab the actual mean values. And again, these are going to be points. So we've got this icon. These are solid points. Limits on the size of the axes. That's just for readability. And then some labels for the names. And since we have standard deviation, we want to add some intervals. And so we can do that by having an upper, I'll call that the upper limit, and the lower limit, UL, LL. And so we take the GSI mean and add the standard deviation, and the GSI mean and subtract the standard deviation to give us the upper level and the lower level. And then the arrows are just basically the way that we plot. It's a function to plot these standard deviations. And here's this X value. It's saying at the X0 and X1, so meaning the lower X and the upper X, we're going to go from 5 to 9. And the Y values, the lower, or the starting point is the upper. And we're having this other point, the lower. And the code 3 means they're capped off like you would for a confidence limit. And the angle's at 90 degrees. And this is just a length in points. And so this is one of the things about R that's a little annoying, is this is a hard way to make standard deviation bars. For folks that use R, you'll probably notice by now I'm only using base R, and I chose that specifically just to keep things as simple as possible. There are other numerous plotting programs. There's a set of programs called ggplot that are packages that you can use in R. They're part of something called the tidyverse that are very, very popular, very common. So what I'm showing you is not the only way to plot. There are many ways, but this is just one. So let's now select all this, Control R to run it. And we see here's our output for the GSIs. And again, the months of the year where we've got 5 through 9, so May through September, and how those GSIs seem to peak during that July value. Even though there is a lot of noise based on the standard deviations, we find that there's a summertime peak. And then we like that plot, so we're going to again come over here, select Control R, and copy and paste it. And that is what I'll cover for spawning periodicity. Are there any questions with those methods?
Silahkan Bapak Ibu kalau ada pertanyaan. You don't have any hand raised yet.
Okay. I'll wait for a sec in case anyone. It's fairly straightforward. I mean, this is just where we've estimated the means, we've calculated the standard deviations, and we've just plotted them. Okay, well.
Also don't have question in the Q&A.
Could a box, so I have a question in the Q&A. Could a box plot be used to look at periodicity? It could. It absolutely could. Yeah, because if you think about let me think about this. The GSIs within each month. So instead of just plotting a mean, you could create, the standard quartile and median-based box plot with outliers and having some of those points. So it's definitely possible and could be an alternative here. And R, you could do that in R also.
We have another question. Silakan, Pak Denny.
Okay, terima kasih Ibu. Professor, can this program show us for multiple spawning periodicity? Thank you.
So meaning multiple, it could, I mean, it really depends on the data, I guess is the answer. Sometimes in certain species, you may see distinct peaks during times of the year. So for example, not for fishes, but I have a collaboration here in Hawaii on limpets. So these are shelled gastropods, and they seem to have a bimodal spawning peak where they'll have at different times of the year. So if you sampled throughout the year for GSIs, you should be able to pick up multiple peaks as they happen. But again, if the data has these multiple peaks overlapping, it then kind of just looks like a single large peak. So it really depends on what kind of information and the frequency that you collect the data.
Oh, oke. Thank you, Profesor. Terima kasih, Ibu. Waktu kami kembalikan.
Terima kasih. Any other question for Pak Eric about not just Pak Eric, Pak Ken as well. From today's training, training hari ini, training yang kemarin tanggal 1 Januari atau mungkin training-training sebelumnya. Jika ada pertanyaan, karena hari ini adalah hari terakhir pelatihan kita, jadi mumpung masih ada waktu, silahkan Bapak Ibu.
Okay. Well, I've got just a couple extra steps here to share. So throughout the training, we have been, let's see, where is my... Throughout the training, we've been writing out to this working directory, and apologize if these are tough to see. I'm going to magnify here. So this is our working directory. Is that, sorry, magnifier's kind of pain. Kim knows that. Okay. So as we've been working along throughout, we've been writing out some of the information that we've had, and I want to share what some of this looks like. Just this list includes a plot of the bimodal output, the fecundity and length data, or sorry, fecundity and length plot, the GSI for females in a table, a plot of the GSI for females. Here's our initial raw data that we have been using. And then we've got our stats in R. This is the code that we've been using in R. The sex ratios are a table that is output. And then the weight length ratio is weight length relationship is a plot. And so if we were to just open one of these, I'll have to share that. So here is output from the female GSI that's now created a CSV with all of that data available. So if you want to use it in another program, or if you have something else that you need to do with this information, you can write it out quite simply like that. For the plots, I've chosen to put these into PDF form, but you can write them out as TIFFs or JPEGs or other vector-based plots. Here's the fecundity at length, the GSI plots, the weight length plots. So all just kind of conveniently output for further use. And then the last one I'll just share is this sex ratio. Which this is a text-based output, if you remember. And so this tells us what the outputs for the sex ratio tests were. Here's the overall sex ratio is 1.08 to 1. We used a chi-squared test as a contingency test. That statistic was 0.88 and the p-value and what our conclusion was from that. And the same kind of information for the operational sex ratio. And so the beauty of doing all this is that, let's go back to our training. I'm going to delete everything in here that we output, except for, oh, hold on, let me close them first. Except for our initial data, our raw data IDLL file, and our stats in R file, our script file. So let's go back to R. And what we can simply do is anywhere in here, I'm going to Control A, and Control A means select all of the text. I'm going to Control A and then Control R. And so now I'm running everything. And it's quite busy. And you see a QQ plot there because there's some extra at the end, but it's quite busy in here. But let's go back over to our training folder. And you can see I've just, at literally the push of a single button, created all of the outputs in an automated fashion. And so if you had data instead of the raw data IDLL in the R, the initial, let's see here. Sorry, go to the top. In the initial input data, right here, if you have your own information, you can bring it in as a CSV there. If it's formatted similarly to the way our data is formatted, you can run all of the exact same analyses. I don't usually suggest doing that because you never know what could have changed, and you might have changed the code yourself. But if you know that it's all correct, you can automatically create all of the output immediately that way. And the work is reproducible, and you know exactly what went into it. You can go back to the code and share it with folks, and it's clear exactly what steps you took to take your analyses and find your results. So that was kind of the last point I wanted to make about that, that it is automated as someone mentioned in the Q&A. And that's one of the beauties of using R, is it's all reproducible and very clear for someone to follow. And with that, I'm going to bring my session to a close.
No, we have one person raising hand, Eric.
Okay.
Pak Agung Abadi, silahkan Pak. Pak Agung. Pak Agung ada pertanyaan? Ya, silahkan. Halo Pak Agung. Okay, the next person is Celia. Celia, silahkan Celia.
Okay, thank you very much, Bu Eni. Okay, it's very interesting, and I have a question about script R. If one day we have a plan to analyze using another model, do you have any suggestion where the website or link for us to download another script? And then, do you have any suggestion for the forum to sharing each other about the problem when we running R? Okay, thank you.
Yeah, so if I understood the question, I think it's if you want to run other models besides what we're presenting, not your own data for these models. Is that correct?
Yes, I mean, if one day we have a plan to analyze, let's say, stock assessment or many things, do you have any suggestion where the website or forum that we can get the another script for analysis? Thank you.
Yeah, okay. It depends on what type of analyses that you want to run. Often, if it's a stock assessment in particular, I suggest that you look at some of the RFMOs when they do a stock assessment. If you look at the document itself, often at the end of the document, there will be appendices at the end that include the software code that they use to run the stock assessment models. And so the approach that you could do is you could look and see how they ran. If, let's say, you want to run a surplus production model and they did it for their assessment, you can look how they used their code and try and replicate their results with their data. And then once you're comfortable with that code, then you could try using your own data to run the models. But they're really, other than stock assessments, there aren't necessarily great forums out there. I think, you know, if you have colleagues that are stock assessment scientists, definitely use them as resources for how to improve, you know, or expand beyond the R models that you know and approaches. But there isn't really a single forum specifically, say, for R stock assessments. If you have just general R questions, there are good online forums for scripting and learning how to do different, you know, just tips and tricks and how to do different analyses and data manipulations. Stack Overflow is one, and there's several others. But is that helpful?
Yes, very helpful. Thank you very much.
Okay. You're welcome.
Oke, Pak Agung, masih ingin bertanya Pak Agung? I guess, ya, Pak Agung silakan Pak. Probably he's having connection problem. There is a question in the Q&A box in Bahasa Indonesia asking how many correct answers have to be done to get through this statistical test. Berapa jawaban harus menurun? I'm not sure what statistical test. Is it the exam or
Probably the exam. Yeah, so the exam for this this module will have 10 questions and you need to pass 9 of them.
Yeah.
And they're randomly chosen from a pool of questions, so they change every time you take it.
I think we are done, Eric. So we are now in the feedback polling before the closing.
Okay, excellent. Thank you so much, folks.
Thank you. Mas Dendi mungkin bisa tolong di running. Bapak Ibu, teman-teman semua, kita sekarang sudah mendekati akhir dari pelatihan secara daring ini yang sudah kita lakukan selama 4 sesi di 4 hari yang berbeda. Kami dari pihak penyelenggara atau panitia sangat berterima kasih kepada Bapak Ibu apabila bisa memberikan feedback atau masukan terkait penyelenggaraan dari pelatihan secara daring ini. Kami akan melaunching ya pollingnya ya. Silahkan Mas Dendi. Pollingnya akan di launching. Bapak Ibu tinggal memilih jawabannya multiple choice.
Maaf Bu Eri, saya nggak bisa mengisi.
Kenapa Ibu tidak bisa mengisi?
Nggak bisa dipilih, tidak aktif itunya.
Oke. Pak Dendi, Mas Dendi bisa tolong bantu Mas Dendi untuk Ibu Husna?
Ya, saya pencet nggak aktif.
Oh. Ini di running lagi sama Mas Dendi nih.
Oke, bisa. Makasih Bu.
Yuk. There is a question, Eric, in the Q&A box asking about the difference on using sum square in Excel and GLM in R. Is it going to be producing the same result or different result?
For the sum of squares?
Yeah, in Excel and in GLM in R.
Let me see well, for which, I guess, the size of maturity tests or which tests?
He didn't ask, he or she didn't ask.
Or models. So you should, for almost all of these models and tests, you should get the same or very similar results. The one that you might have some different results are the size of maturity models, because for that you're using solver in Excel. And so that's an estimation. It's a non-deterministic estimation of parameter values. So they might be a little different between the two. But I think if the question is, should you be getting the same results between R and Excel, the answer is yes. Okay. And that's part of why we present both approaches is when you start to get into this field and do more analysis, it's nice to have multiple methods to give you a result to evaluate whether or not one is different than the other or correct. So this should be the same here.
Yeah, I think he's asking about predicting L50 and L95. Okay. Great. So pollingnya masih berlanjut ya. The polling is still ongoing. I guess there was some technical glitch a while ago that some people can actually answer the poll. And another question. Oh, terima kasih. Thank you, Pak. Ya. Kami dari pihak panitia akan mengcapture foto Bapak Ibu, dan kalau Bapak Ibu bersedia untuk diambil gambarnya, bisa tolong nyalakan kameranya untuk nanti kami membuat laporan. Kalau tidak bersedia tidak apa, silakan ditutup kameranya. Terima kasih. Nanti di akhir pada saat Zoom ini tutup juga nanti akan ada survei 1 pertanyaan yang Bapak Ibu bisa isi di situ. Yang tertulis open end question.
Bu Eni?
Ya.
Yeah, while waiting the survey, I would like to say something. Is it okay?
Please, Pak.
Thank you, Bu Eni, for this opportunity. I would like to deliver our appreciation and also thanks for the team Pak Eric, Pak Ken, Pak Ros, for your kindness and providing your time and also your expertise to share with us your knowledge. I think this is very useful for us. Thanks for that effort. So I feel that the training is useful and then easy and can to follow to and then to understand. Of course, increase our knowledge and along with that, I also would like to thanks to Bu Eni, the STA team, Pak Dendi. Maybe I cannot mention this one by one. And also the most important thing is thanks for all participants, because without participants, this training will not be worth it. So the participant also very important for this. So thank you, Bu Eni. That's all that I would like to say. Thank you.
Terima kasih Pak Fayakun. Ini ada pertanyaan dari Pak Supradianto. Pak Supradianto, apa ini Zoom-nya aja glitch ya? Barangkali tidak. Oke. Gimana Mas Dendi?
I think we have the maximum number of people involved in the polling, but we are not showing the result for the survey, for this enough training survey.
So, I mean, the result, is it the training, apa namanya ini, survey, is it completed or is it still people giving answer?
Yep, I don't see any movement on the numbers, so I guess we make it complete.
Yeah, we can close it, yeah, Mas.
Yeah, thank you.
Okay, great. So, okay, so I think now we have reached to the end of the event, ya. Oh, Bapak Ibu, kita sudah tiba di akhir acara. So I think now Pak Eric will deliver closing remark, ya Pak Eric. You're muted.
I was gonna say everyone's so excited for the group photo. I almost don't want to talk. Just get to it. So this concludes the second module in the entire training course on fish reproductive analysis. We are extremely happy that there were over 800 registrants and almost 300 to 400 active participants from Indonesian government, academia, NGOs, and the public. On behalf of the other instructors, we thank you for participating in the two modules and hope that you learn new information about fish reproduction. To recap, the first module provided an overview of the anatomy, development, and classification of fish reproductive structures. We described the characteristics of female and male gonads, ovotestes, and other structures, and presented microphotographs of gonad and other tissue sections as examples during that training. We also addressed questions from course participants and provided time to attempt the online exam. Most of the information and resources that are presented are from our technical report, the standard operating procedure for histology-based rapid reproductive analysis of tropical fishes, for which we provided online links. In the coming weeks, a translated version of the SOP in Bahasa Indonesia will also be made available online. And during this second module, we introduced the statistical analysis and plotting of reproductive data, including methods in Excel and R used to calculate size of maturity, sex ratios, fecundity, bimodal size distributions, spawning periodicity, and weight length relationships. We included an example data set to demonstrate the different analysis and plotting methods and provided all the materials to participants. And just a reminder, for both modules, the exams will be offered online for the rest of the year and can be taken as many times as necessary to achieve a passing grade. Once an exam is passed, the participant will receive a certificate of completion issued online from the University of Hawaii. And finally, we offer our thanks to Pak Sharif for opening the training with his welcome introduction and for the support and participation of the Ministry of Marine Affairs and Fisheries, KKP. We also thank our moderator, Ibu Eni Buhari, the technical coordination for the webinars by Pak Dendi Windy, the support staff, Pak David Kuntel and Ibu Fina Fitriani, and our translation team of Ibu Rika and Ibu Septi. This training is part of the supplemental technical assistance to the USAIDC project managed by Tetra Tech and funded by the Walton Family Foundation. And most of all, we thank you again for actively participating in the course and look forward to potentially more courses in the future. Thank you.
Terima kasih Eric for your closing, and juga terima kasih Bapak Ibu atas partisipasinya. I think I'm echoing. Selama ini 4 hari acara seri pelatihan secara online. Sekedar informasi bahwa sebagian dari Bapak Ibu kontak kami menanyakan tentang apakah ijazahnya atau sertifikatnya itu ada jumlah jam pelatihan. Jawaban kami tidak, di situ tidak ditulis jumlah jam pelatihan selama pelatihan ini, tapi nanti akan kami kirim poster yang dulu mungkin Bapak Ibu pernah lihat, di situ ada jamnya. Jadi nanti akan kirim poster itu ke email ke Bapak Ibu semua jika memang dibutuhkan informasi jam tersebut. Kemudian rekaman dari acara ini selama 4 sesi akan dilakukan oleh panitia nanti, dan kalau bisa juga akan dikasih subtitle dalam bahasa Indonesia atau bahasa Inggris, dan apabila sudah selesai prosesnya akan kami upload ke sebuah akun YouTube. Akun YouTube mana nanti akan kami informasikan lebih lanjut melalui email ke Bapak Ibu sekalian. Demikian sekedar informasi terakhir dari kami. Sebelum dan sesudahnya mohon maaf jika ada kekurangan, technical glitch dan sebagainya di dalam pelaksanaan acara ini. Saya Eni Buhari pamit undur diri untuk hari ini. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you.
Thank
Terima
you.
kasih Bu Eni. Thank you.
Have a great time.
Thank you.
Hope everybody enjoy it.
Turn on your camera and wave so that I can have.
Thank you.
Thank you.
Terima kasih.
Terima kasih sama-sama.
Thank you to panelists.
Terima kasih banyak.
Terima kasih banyak.
Terima kasih Ibu semuanya.
Thank you very much.
Terima kasih, matur nuwun.
Terima kasih semuanya. Ini dari seluruh banyak provinsi dari Indonesia dan juga dari luar negeri ini rupanya.
Semoga sehat semuanya.
Terima kasih. Selamat siang, selamat sore, selamat malam.
Iya, selamat siang nih. Kita masih siang. Pamit dulu Ibu semua para profesor. Thank you.
Ya, silakan.